Well, I love finding out something new about the world that nobody has ever known before. That's great. And I also love solving puzzles, and chemistry is often solving puzzles like working out the structure of an unknown compound from the NMR data or other data like that. So I love that aspect as well. We research into the biosynthesis of natural products, and we also use bioorthogonal chemistry to study the sugars on the outside of cancer cells. Major impacts were discovering the full biosynthetic pathway to vitamin B12, which was quite an undertaking, and also to the red pigment prodigiosin. Those have both had big impact on the scientific community. Currently, we've got a method that we helped develop for detecting the response of tumours in the first few days after treatment, and that is going forward into clinical trials, and that could really make a big difference to treatment of cancer if it's successful. Often the impact comes from things you don't really expect. My most cited paper was on organocatalysis, and the reason it's been cited so much is not because of the results of that paper, but because the synthesis of one of the catalysts we used is the same method that everyone has followed ever since who's been in the field. So we get lots of citations, and I really didn't expect it for that paper. 3 things we're working on or want to work on. One is the very last bit of that biosynthesis of prodigiosin that I mentioned, the red pigment, because there's just one enzymic step that we haven't been able to prove definitely yet. It's an enzyme called PIG-E, and we're pretty sure it is bifunctional, reducing a thioester to an aldehyde and then transaminating that to an amine, but we haven't been able to prove it directly, and we'd love to do that and put the last piece in the jigsaw of prodigiosin biosynthesis in place. Another biosynthetic project, well, it's structure determination and biosynthesis. It's a compound that we're currently calling metilamide, but we don't know the whole structure of it. We know 2/3 of the structure of it, and it's most remarkable, the 2/3 that we know. It has 5 dehydroalanine residues in a row put in there by it seems, one peptide synthetase being iterative 5 times over, and that's completely unheard of. We want to develop bioorthogonal reagents that react faster and are more water soluble, because those are both limitations to the method, and use these to image the incorporation of 2 different sugars into the cell surface glycans of the cancer cells. And we're hoping that will be a way of diagnosing how invasive the tumour is. The important thing is to do novel work, finding out new things about the world and publish it, and don't worry too much about the impact, it'll come.